Hi guys. Today we are going to talk about TGF-beta receptor. So the context comes from this paper where we are talking about the expanding role of small molecules in immuno-oncology. So the advent of immune checkpoint inhibition using antibodies against PD-1 and its ligand has prompted substantial efforts to develop complementary drugs. Although most of these are antibodies, they are increasing interest in small molecule immune-oncology drugs that address the intracellular pathways. Right. So, if we look at this image from this paper, we can clearly see that TGF beta receptors are an important component. If you look at this part, and if you also look at this part where they basically activate M1 and NK cells, which are involved in immuno-oncology, and they also are involved in activation of DC cells, right, which are involved in cancer pathways. So TGF beta is a very complex pathway where it can act as tumor promoter as well as tumor suppressor in some cases, right? So and this paper clearly says that this TGF beta signaling is important in immunity and cancer, right? Perturbations in TGF beta signaling underlie inflammatory disease and promote tumor emergence. So, if we inhibit TGF beta receptor, it could lead to the tumor suppression, right? So, so the point is, if we look at, there are lots of TGF beta receptors which are already there. So, can we use our platform, our drug discovery platform, to find novel inhibitors, right? So we go to our platform and we click here to generate new molecules, right? So basically you can so what it takes as input is a CSV file, which I will just show you here. So this is the CSV file which it takes. It is on There is also a sample file you can see before uploading. So these are the files, and this is the PCI50 value, right? Which is just the IC50 value in nanomolar, right? So that is the concentration which is needed to inhibit 50% of this protein, right? So an important thing to note is if you look at this data, so this data is usually is comes from experiment where they have done a large screen of against this protein TGF beta receptor, and then for each chemical molecule you have the IC50, right? But this can come from large screens also. This can come from ChEMBL database also, which I will come in a short while. So the important thing to note is these values are not logs. These values are linearly spaced, right? So they just go in tens, right? So they multiply by in tens, right? So what is needed is basically to take the log of this, right? Minus 10 log of this, right? So that way, what you have is a linearly spaced data, right? And then, so usually what you want to do is you want to find the molecules which have a low value of PIC50, right? But if I take the minus log of 10, you would need high values of this number, right? So we should keep that in mind. So let me just upload a file, right? This I have uploaded. So just we have to wait a couple of minutes to get the AI model results, right? So basically what happens is in this model is, so from the data the AI makes a AI model which basically predicts for any new molecule what is the IC50, right? And then it uses recurrent neural networks which and other complicated networks to basically train the AI model to find novel inhibitors which are basically better than these, right? So for example, the minimum we had was 0.55 nanomolar inhibitor. So but the problem with this is a known molecule, right? If you find ChEMBL, you will find, so you can't patent this molecule, right? So the point of this is, can you get a better AI model, better molecules which are novel and lower than this value consideration, right? So I will, while these results are coming out, so I will explain you how did I got this data, right? So as I was telling you, this data was obtained from ChEMBL. So we go to ChEMBL, we write TGF-beta receptor type 1, we enter, right? And then just a sec, yes. So we have three targets here, right? So this target has a maximum 3612 activities, right? Maximum compounds against, and this, so we click this, right? And then we take the IC50 values, which is 1681 molecules, right? So as you can see, these are the molecules and these are the IC50 values in nanomole, right? So we can download, just a second when this gets loaded. So we can, so we can download this result, and when we so the results looks like this. So this is the data. So we take the negative log of 10, and then we upload this column and this column in the this file. So these results are out. So basically the r square value, which is the correlation coefficient of the model, right? So in predicting unknown set of molecules is 0.53, 0.59, 0.52 against different set of training and testing, right? So as mentioned here, only models with good R square values greater than zero point 0.5 will predict accurately new molecules, right? So, for example, if I would have taken these two rows, right, which is not log space, right, which is like linear, which is multiplying in terms of 10, right? So, so what would have happened is this R square value would have been very low, right? I'm not showing it, but you can check it out, right? For low R square values, try again taking log 10 of the data. For good R square values, proceed accordingly, right? So you can even contact us, you can mail us to improve these scores. But now, since our square value is good, we can go ahead. As I mentioned that IC50 we want lower molecules, but if I'm taking negative log of IC50, we want a higher score molecules, right? So I go to higher score, submit, task for password, right? So you'll get results in your email. Please wait till the job is finished. This job will finish in 12 to 15 hours, right? So we can't wait for 12 to 15 hours. I'll just show you the results for which I have already collected. So just a sec. These are the basically scores. So distribution scores for untrained and trained molecules. You need not. These are for high advanced training. So basically, in short while. So the orange is the trained one, blue is the untrained one. So before we do any model training, which took 12 hours. So this was the distribution of molecules and oranges. So basically, you can see that the distribution has shifted towards right because we were looking for high scores. Distribution was not so much shifted towards right. This depends on protein to protein, right? But what is interesting for us is we can download the results, right? So I have already downloaded the result and I can show you the result. So these are basically the molecules. So these are the molecules. This is biased. So as we can see that as we can see that I have sorted it based on the IC50 value. So we can see that the lowest molecule, lowest concentration molecule which we got was 7.44 nanomolar. Right. So if you look at the original data, the minimum was 0.55. Right. So this there are lots of molecules less than 7 nanomolar also. But the benefit of this is even though this is not the minimum we found, it depends on protein to protein, as I said, and the model training. But what we got, so these molecules are novel molecules, right? If we, I can't show you the exact structure of this because we are working on it for patent and etc. So these molecules are novel and patentable. So one can always look at first 10 molecules up to 27 nanomolar, for example, and synthesize the molecules in experiment and test it, right, against cancer activity, right? So in a sense, what I have told you here is immune-oncology small molecules are currently a hot topic. This is a August 2022 paper. And if we look at this, TGF-beta inhibitors are actually hot topic. So we use this application to first we go to ChEMBL, download the data, upload it here, and then we, as I showed you, we can get good molecules which are nanomolar affinity molecules, right? So thanks for this. If you have any questions, you can ask in comments. Thank you.